Hi everyone, I'm Savannah.
And I'm Christina, and today we're going to be explaining the nervous system.
To begin, we'll be discussing the three overlapping functions of the nervous system. These include sensory input, integration, and motor output. In the process of sensory input, this is where we take information in. Once the information comes in, our brain interprets and integrates what we are seeing and decides what to do with it. Finally, our response to the stimulus is shown through the process of motor output. This is the stage in which we act.
Now we will transition into the organization of the nervous system. The nervous system is made up of two different systems, the central nervous system and the peripheral nervous system. First, let's talk about the central nervous system. The central nervous system is made up of the brain and the spinal cord. It is to be noted that both structures have different functions. Your brain handles your thoughts, major senses, and what you do, while your spinal cord is responsible for your reflexes and your response to different stimuli.
Now onto the peripheral nervous system. The peripheral nervous system is pretty much everything else. This system is divided into sensory division and motor division. In sensory division, we have afferent information coming in. Let's start with the somatic afferents. This is where we have sensory information coming in from the skin, muscles, and joints. Moving on to visceral afferents, this is where we have sensory information coming in from our internal organs. Lastly, we have special sensory afferents. This is where we receive sensory information from our senses, including our eyes, ears, nose, and mouth.
Now we'll discuss motor division. This is where we have efferent information going out. Let's start with the somatic nervous system. These actions are mostly voluntary and work in a way of how we think and carry out a response. Next is the autonomic nervous system. These actions are mostly involuntary and include the movement of cardiac muscle and smooth muscle. The autonomic nervous system includes the parasympathetic and the sympathetic nervous system. Our sympathetic nervous system is responsible for our fight or flight response, especially in stressful situations, while our parasympathetic nervous system is responsible for conservation of energy and slowing our heart rate and increased activity in digestion. Now on to the histology of the nervous system. We can divide this section into two categories, these being our supporting cells, also known as neuroglia, and our neurons, which are known as nerve cells.
In the CNS, there are four types of neuroglia. The first one we'll be discussing are the astrocytes. These cells help control the chemical environment around a neuron and specialize in the formation of the blood-brain barrier. The next glial cell we'll be discussing is the microglia. These cells are known for being macrophages. They are the cells that consume cellular waste. Moving on to the ependymal cells, these cells line the CSF-filled ventricles in the brain and spinal cord. They produce cerebrospinal fluid. Lastly, we have the oligodendrocytes. These cells form the myelin sheath and provide support and insulation to axons in the CNS.
Now on to the two types of neuroglia in the PNS. The first being satellite cells. These cells cover the surface of nerve cell bodies and provide support to the neurons. The last type of neuroglial cell in the PNS are Schwann cells, also known as neurolemocytes. These cells form the myelin sheath and provide insulation in the PNS. These are not to be confused with the oligodendrocytes, which are found in the CNS.
Now we will explain the neurons. Let's start with the basics to get a better understanding. To begin, we'll cover the functional characteristics of neurons as well as the neuron's anatomy. There are four main functional characteristics of neurons that include their ability to conduct impulses. This is where our neurons carry electrical messages across the body. Next is their extreme longevity, meaning they can basically last a lifetime. They are amitotic, in which means they do not divide. And lastly, have a high metabolic rate and require a lot of oxygen and glucose.
Next, we have the structural classification of neurons. These are divided into three different parts or types of neurons. The first part is multipolar neurons, which have more than two processes sticking out of the cell bodies. The next one is bipolar neurons, which have two processes that stick out of the cell body. Lastly, we have the unipolar neuron, which only has one process that sticks out of the cell body.
Now onto the anatomy of a neuron. Each of our neurons have a cell body in which encase many important features. The first being Nissl bodies. These structures make proteins. Next, we have the neurofibrils and the neurofilaments. These maintain the cell's shape. We also have what's called lipofuscin, which is the pigment found in our nerve cells. Next, we have the axon hillock. This structure is very important, for this is where the generation of action potential takes place. We have a collection of cell bodies in the CNS, which is called the nuclei, and a collection of cell bodies in the PNS, which is referred to as the ganglia.
Next, we have bundles of processes, also known as axons. In the CNS, they are referred to as tracts, and in the PNS are referred to as nerves. Now on to dendrites, which act as listeners of the neuron. They have a receptive region where they receive incoming stimuli. They also have a graded potential, which are charges in the membrane. Lastly, we have the axon, which consists of many features, the first being the axolemma. This is where a message is carried. We have the terminal branches, which are located toward the end of the neuron, and lastly, we have the axon terminal, which is a secretory component of the neuron.
Now we'll talk about the transport along the axon. This can be distinguished between anterograde transport and retrograde transport. Anterograde transport is transport towards the axon terminals, while retrograde transport is transport back towards the cell body. It is to be noted that there are different types of axons, one being myelinated and the other being unmyelinated. Myelinated axons have a myelin sheath. This structure provides insulation and speeds impulses within the axon. The myelin sheath can be made up of two different types of cells. In the PNS, the myelin sheath is made up of Schwann cells, while in the CNS, the myelin sheath is made up of oligodendrocytes. When learning about the brain, we can observe two different types of tissue in the CNS. There's white matter, which is made up of myelinated axons, and gray matter, which is made up of unmyelinated axons. Lastly, we have what's called the nodes of Ranvier. These are the spaces in between the Schwann cells. These are quite significant, for they help with saltatory conduction. This is where an impulse jumps from node to node in an axon.
Now we will discuss neurophysiology. First, let's begin with electrochemical gradients. In an electrochemical gradient, there are more chemical charges on one side of the axon than the other. In order to balance out different charges, we must have different channels and gates to let certain ions in and out of the axon. First, let's begin with passive leakage channels. These channels are open all the time and let different ions in and out of the axon. On the other hand, we have active gated channels, in which are opened by an electrical charge or a neurotransmitter. In other words, chemically gated channels open through neurotransmitters, while voltage-gated channels are open due to an electrical charge. Now let's transition into what's called the resting membrane potential. Resting membrane potential is broken down into three different characteristics, the first being unequal distribution of ions across the plasma membrane. Next is the selective permeability of plasma membrane. This includes keeping sodium out and potassium in. Lastly, we have the sodium-potassium pump, which keeps the sodium and the potassium at unequal distribution and uses ATP to pump 3 sodium outside of the membrane and 2 potassium inside.
Now we will move on to membrane potentials that act as signals. These include depolarization, which is the reduction in membrane potential, repolarization, which gets farther away from zero, and finally hyperpolarization, which is when the membrane is greater than -70 millivolts. Next are different types of signals, such as graded potentials and action potentials. Graded potentials are found in dendrites of the cell body and are short-term messages, whereas action potentials have the same strength the whole way through and are only carried in the axon.
Next, we will explain the process of the generation of an action potential. First, let's start with the resting state where the voltage gate channels are closed. At this state, there is a positive charge on the outside and a negative charge on the inside. Next, we have depolarization, which is where the sodium gates open. Sodium rushes in, resulting in a change in positive charge on the inside and a negative charge on the outside. This is where we have hit threshold. Moving on, we have repolarization. This is where we have potassium rushing out, resulting in a decrease in sodium permeability and an increase in potassium permeability. Lastly, we have hyperpolarization. This is where the charge is lower than negative 70 millivolts.
Next, there are two types of refractory periods. Refractory periods make it almost impossible to fire a neuron. The first type is absolute refractory period. This is where it's almost impossible to fire a neuron again until another action potential is generated. The second type is called relative refractory period. This is where it's more difficult to fire a neuron again. However, it is possible, you just need a strong stimulus. Moving on to the synapse, this is where a neuron connects to another neuron. The neuron before the synapse is the presynaptic neuron, while the neuron after the synapse is called the postsynaptic neuron.
Now we will discuss the two types of synapses. The first being electrical synapse, which is another term for gap junctions. This allows waves of ions to communicate. Secondly, we have the chemical synapses. These use neurotransmitters to either open or close ion channels. Now we have the organization of the synapse, which consists of the axonal terminal of the presynaptic neuron, the synaptic cleft, which is a space filled with mostly water, and receptors of the postsynaptic neuron. Thirdly, we have the information that transfers across the chemical synapse, also known as excitatory. On to the termination of neurotransmitter effects, and finally the synaptic delay, which is the amount of time it takes to cross a synapse.
Next, we will discuss the postsynaptic potentials and synaptic integration. With postsynaptic potentials, we have two different kinds. These include excitatory synapses, or EPSPS, or inhibitory synapses, or IPSPS. Excitatory synapses depolarize the axon hillock and make it more likely to fire the action potential. Inhibitory synapses hyperpolarize the axon hillock and make it less likely to fire an action potential. Moving on to integration and modification of synaptic events, first we have the summation of postsynaptic neurons. There are two kinds of summations, known as temporal summation and spatial summation. Temporal summation occurs when graded potentials happen close together in time. Spatial summation occurs when graded potentials come in from different directions. Next, we have synaptic potentiation, which is also known as the warming of the synapse. Lastly, with presynaptic inhibition, this is when one neuron inhibits another neuron. In neuromodulation, other modulations affect when things fire, for example, hormones.
And that concludes our explanation of the nervous system.
Thanks for watching!